Okay, so in here I gave examples of there is a mutation in the gene. So this is some gene that encodes an enzyme. That's the enzyme that does some job in the cell. Sometimes the mutation is not on the gene. It is in the regulatory sequence. So it's in the DNA sequence upstream from the gene. So like, let's say you have the gene itself encodes a functional protein. But if there is a sequence here to the left, so upstream of the gene, and that could affect transcription of this gene, and that's how you affect the phenotype by not making like a functional and non-functional subunits, but by making the functional enzyme, but just not a lot of it. So let's look into that. We are going to look at haploinsufficiency because of a change in transcription, which is, okay, this is caused by mutation in the transcription factors. Actually, I'll go over one and then I'll come back to this. There you go. So that slide is going to show up again. I just wanted to talk to you about the mutation in the promoter region. So here, if we change how much transcription is made, then you would change the phenotype. Here you have a wild type snapdragon, and there's a bunch of red pigment. This thing here, the boxes, the pink boxes, they represent the exons of that chalcone gene. So this is to show you why did I insist on that gene prediction software. I wanted you guys to know the pieces of a gene, the parts of a gene, so that now this would be easier to understand. So here is some reminders. These guys are the exons. And you're like, okay, what is an exon? It's the DNA sequence that after this is transcribed, those parts will be translated. These parts here will not be translated. So there is transcription of this. There's transcription of all of this from here to here. But then this part will be translated, and then this will not, this will not. If this is your last exon, your stop codon is here. This part is transcribed but not translated. What's that one called? So this part here between the no, the intron is this and this. So here, let's see. This is a start codon. So this encodes your methionine. That's your first ATG. And then this is one of the stop codons, right? So this is your 3' UTR. Okay, the introns are the DNA in between exons. But after this stop codon, it's a UTR. And before our start codon is also UTR. So this is the 5' UTR, 3' UTR. And then in the exam, when I said, what is a gene? What makes a gene different from non-genes? So if this is the gene from here to here, that means that this DNA sequence appears in the cell as RNA, meaning there's an RNA molecule made from transcription of this part. So I was looking for gene is a DNA sequence that is transcribed. Okay, so those are the exons. These guys are the introns. And then over here, so it's really like upstream of here. People draw this little arrow like that to say this is where transcription starts. So I should have put this arrow a little bit above it. Okay, the promoter is the upstream sequence to the beginning of transcription. That are parts of the promoter. So some parts of the promoter are part of the mRNA. But think about the promoter as the DNA sequence upstream of the gene that is controlling how many mRNA molecules of this get made. And so now you're going to learn how does it do that? Maybe like in a few slides from now. But so far so good? All right, so that's the gene. Here's what the promoter is, what controls how much of this gene gets made. In this case here, the wild type, a lot of the enzyme gets made. So then you have this dark, colorful flower. The wild type is encodes a functional enzyme. And then people said, okay, let's call it the amount of expression of that gene 1. Okay, so this is like when 100% of this gene is made, and then there's 100% action of that gene, we're gonna say 1. So that's 100% of action. All right, so this is as pink as you could get. Okay, now that gets a little complicated. So maybe let's just go to this part and then I'll show you this next thing. Okay, this thing here is a transposon. It's called TEM3. So I put there, a TEM3 is a transposable element. That's just the name of that one specifically. And so this chunk of DNA gets inserted here. And you're like, okay, but since the transposon is not inserted in the exon, maybe it doesn't do anything. Or maybe it does. So if the transposon got inserted in the exons, then it very likely impacts the phenotype, because the protein that gets made is all screwy, and maybe it can't do the job that it normally does. And then you hear like, okay, the transposon is not in the exons, it's in the promoter. And then maybe you're like, oh, okay, you said if it inserts itself in the exons, then it changes things. How about if it inserts itself in the introns? Okay, you're going to see an example in homework when that happens, and maybe it changes the phenotype, maybe it doesn't. Okay, so let me do a poll just to see if you guys are here. If the transposon inserts itself on the promoter, which is this DNA sequence upstream of the start of transcription, do you think it will impact the phenotype? So that's the poll I'm going to write now.